Kia ora! I'm a small scientist, and today I'm going to talk to you about environmental DNA in Antarctica. Marine ecologists love the Earth. Like, a lot. One of the questions we have to answer for a variety of reasons is who lives where in the ocean. In Antarctica, that can be pretty hard. It's cold, icy, and not to mention dark. We don't know very much about who lives under the water. But not to worry, science has got a way. All living things have cells, and in those cells are DNA that's unique to them. As an individual moves about, it naturally sheds cells, leaving DNA in the space around it. This is called environmental DNA, or eDNA for short. Scientists can take a sample of water and put it through a PCR machine, which can tell us whose DNA was in the water. This is a pretty new method, and it's pretty cool because it means that we can figure out what species lives somewhere without actually seeing them. Unfortunately, we can't sample the entire ocean. It's way too big. That's where ecology comes in. We know that sea sponges can filter up to 10,000 litres of seawater a day. That's a really efficient way to collect cells. When we sample these sponges, we get a lot more eDNA from a much larger area. This is pretty cool, and recently, scientists at the University of Otago had a really good idea. NIWA has a lot of old sea sponges preserved from Antarctica. They thought, hey, it would be super helpful if we could take eDNA from these old sponges. So, maybe we could learn about what species were present in Antarctica in the past. They had two questions. Firstly, is the old DNA still there? DNA in the marine environment degrades very quickly, so it'd be good to know if the eDNA was preserved with the sponge. They also wanted to know if different sampling types preserved different eDNA. They looked at sponges that had been frozen, dried, or submerged in ethanol. They took samples and ran them through that PCR machine. And they found Antarctic fish DNA in all three types of sample. So the answer to the first question is yes. The scientists suggested we call this historical environmental DNA, or eDNA for short. They also found that frozen samples had different fish eDNA to those that had been submerged in ethanol or dried. All of this information is really important for future use. To summarize, we can use old sponge samples to learn about what fish species used to live in Antarctica through environmental DNA. Awesome! If you'd like to learn more, please check out the credits or the links in the description. Thanks!